Good day to Prof. Edmund. Today we are going to present about COVID-19. COVID-19 is a highly contagious viral disease caused by SARS-CoV-2. It can cause symptoms ranging from mild to fatal, especially in vulnerable individuals. At the start of pandemic, treatment relied on supportive care, since there were no specific antivirus or vaccine. But for now, we have effective vaccine that significantly reduce severe outcome. The virus emerged in Wuhan, China in late 2019. It rapidly spread worldwide, causing global disruption and prompting WHO to declare a global emergency. It likely originated in bats, but the exact transmission to humans remains unclear. Understanding the biology of virus is critical. It helps in create accurate tests, effective treatment, and vaccine. It also guides public health efforts to prevent future outbreaks.
Now let's look at how SARS-CoV-2 is built and how it enters our cell. SARS-CoV-2 is an enveloped virus with a single-stranded RNA genome at its core. Surrounding this RNA is a lipid envelope, which is a fatty layer that protects the virus and helps it fuse with the host cell. Embedded within this envelope are three main structural proteins which are known as the spike protein, membrane protein, and envelope protein. As for the entry mechanism, the infection begins with the spike protein binds to the S2 receptor, which are commonly found on the surface of the human cells. After this binding, the virus can enter the cells in two ways. One, by membrane fusion, where the virus fuses directly with the host membrane, or two, by endocytosis, where the host cell engulfs the virus. Now once inside, the viral RNA is released, allowing the virus to hijack the cell's machinery and make more copies of itself.
After enter and release of viral RNA, host ribosomes translate viral RNA into polyproteins, which will be cleaved and form replication complex. The replication complex replicates the viral RNA and produce subgenomic RNAs to make the structural proteins. Structural proteins are made and new viral particles are assembled in the Golgi apparatus. These new viruses are transferred to the cell surface and released through exocytosis to infect other cells. Innate immune response is the first-line defense against COVID-19. The virus enters host cell via S2 triggering recognition by PRR. This activates signaling pathway leading to release of cytokines and type 1 interferon which can cause apoptosis. In severe cases, cytokine storm can occur. It can cause tissue damage or organ failure. Adaptive immune response is the second-line defense against COVID-19. It involves cytotoxic T cells that kill infected cells and B cells that produce antibodies. In mild to moderate cases, this response is strong and long-lasting. In severe cases, patients often experience lymphopenia with low levels of B and T cells.
For COVID-19 symptoms can range from mild to severe. Most individuals experience fever, cough, fatigue, and loss of taste or smell. In severe cases, the infection can lead to pneumonia, respiratory failure, or acute respiratory distress syndrome. After an incubation period of about 5 days, some patients worse by the second week, around day 8. Severe symptoms may develop, and by day 16, critical complications like cardiac injury or multi-organ failure can occur. Older adults and individuals with underlying conditions such as diabetes or heart disease are at greater risk of poor outcome. Because of these serious complications, scientists focus on preventing infection earlier. One of the most effective results of this effort is the development of mRNA vaccine.
So the mRNA vaccine is using synthetic messenger RNA to instruct the cell and to make viral protein. This will be focused on the spike protein as the blueprint. So because of mRNA is fragile and easily degrades, hence it will be transported using enclosed lipid nanoparticles. The mechanism of vaccine first is the injection of lipid nanoparticles which contain mRNA into human body, usually the upper arm of the muscle. The lipid nanoparticles will transport to a nearby muscle cell or immune cell. After the transportation to the cell, the ribosome will read the mRNA and to produce spike protein. Synthesis of spike protein activates immune system to recognize as immune memory. This vaccine is fast development as it does not require growing of virus. This vaccine technology is quickly become important tools in the future.
mRNA vaccines developed during the COVID-19 pandemic, such as Comirnaty and Spikevax, showed over 90% efficacy in early clinical trials and significantly reduced severe illness, hospitalization, and death worldwide. Their rapid development and FDA approval highlight a swift global response and validated decades of research in mRNA technology, marking a major breakthrough. The technology's flexibility also allows for quick adaptation to target new variants. The future of mRNA vaccines is highly promising. Their speed and adaptability make them ideal for future pandemics. Beyond COVID-19, mRNA vaccines are being explored for cancer, influenza, and Zika virus. Innovations like self-amplifying mRNA and combination vaccines may lower doses and expand protection. However, issues like cold storage, long-term safety, and access in low-income countries still need to be addressed.